Hi, everyone.
Hello, so we're back for another workshop. It's so exciting.
It is. It is. Wow, I guess the week's passing so fast, and then it's Friday again, and I also feel excited to start a new workshop. So thank you, everyone, to be here. I'm Felipe Matias,
I'm Claire Gahagan.
and today we have a new workshop for the PhenomeForce workshop series. And as usually, we start with our introduction and explaining about our project. Let me share here. Oop, there we go.
Excellent. Yeah, so we are PhenomeForce. We're a learning community. We're trying to make plant phenomics for everyone. So our main goals are to connect people, empower early career researchers, build up a library of online resources for phenomics, and provide free trainings. So that's who we are. And if you want to know more about us or find past workshops, our website is PhenomeForce GitHub. The link is there. It'll also be in the video description. We have a Twitter account. Hopefully you found the YouTube if you're watching this video. And we can be contacted by email as well. We also have a Slack page, and that's where there's a lot of communication and chatting. We also have some details about those Friday sessions. And if you'd like to be a part of that Slack group, then you can just send us an email and we'll add you to the workspace.
Yeah, awesome. Yeah, just remember here, we do have the section for job offers. So if you have a new position in your lab, you also can contact us or send your message right here. And let's communicate, right, Claire?
Yeah, of course. It's a great way to get to know people and get the job offers out into the community.
Yeah, perfect. Mainly for us early careers that is looking for positions. Well, we do have our YouTube, and we had the season that we did last year on 2020. We had already 10 workshops from last year, and the workshop from the second season that we had last week, and was really great. So please go there, check the other workshops, have your time to learn. And if you have any idea or if you want to share the work that you guys have been doing in your lab, please communicate with us. We can try to schedule a workshop for you too. So our main idea, as Claire was saying, is communicate and share our knowledge. And also, if you're new here, please subscribe here at the channel and keep following what we are doing. Every time when you go live, if you are subscribed, you're gonna receive a message. So that's nice for you keeping following our work. And this year we have this Fried Hands-On workshop series 2 or 2021, and our main focus now is using low-cost phenotyping platforms. And low-cost phenotype platforms can be interpreted in a different ways, mainly, for example, building a platform or using and finding data online to do your research. So mainly, actually, the workshop today, we're going to talk about these, using the data that is already available over there. We are going live every Friday, starting last week, April 9, at 10 a.m. Central Time, okay? And then we have this wonderful group here that is helping put all these pieces together. Anahita Mahano, myself, Claire, Noah. So feel free to contact us in our social media as well. The workshop today is gonna be on R, and we did send a message talking about the binder, but unfortunately we do not put the data, have time to put the data exactly inside the binder. So today we recommend to use your own computer, which is gonna be great as well. And if you go here in the description in this video, just click description here. You're gonna see there all information that you need for this workshop. So all packages that you need to install, and also the link to download the data for the workshop today. Those are the 3 packages that we need. Okay. So with this, I wanna introduce the next workshop series, next workshop today, which is using TERRA REF high throughput sensor collected plant data with R. And we have the amazing opportunity to have Kristina and David from University of Arizona to share and stay with us this morning. Okay, so let's invite them. Hi everyone.
Oh, good morning. Thanks so much for having us.
Thank you for coming.
Yeah, it's a great opportunity to talk about our work. So I'll go ahead and I'm going to give a quick introduction to the TERRA REF project, and then Kristina is going to lead the hands-on workshop part. So I'll share my screen now and give the presentation. So the data set that we're going to be talking about today, it's been published on Dryad. As you all can see, there is a very long list of authors, many collaborators, and a lot of work went into running this project, and all of the work that went into preparing the data that we're going to be using today. And as you will learn, there's going to be a very large amount of data, and we're just going to kind of look at a few examples of what's available. And I'll first tell you a little bit about what we have and why we're doing what we're doing. So first, as Felipe mentioned, this is a very expensive phenotyping machine. As far as we know, it's the largest agricultural robot in the world. When we put it on the ground in about 2016, the idea was that we would deploy cutting-edge sensors that were available and scan the field at an unprecedented resolution. Most of the sensors collect data at approximately 1 millimeter resolution spatially. And like the hyperspectral camera, it's smaller than a nanometer spectral resolution. I'll tell you a little bit about the sensors in a second. But while it's an expensive system, part of the justification was that rather than having a funding agency purchase many different sets of sensors from many different sets of groups when it was unclear how the sensor or the sensor data could be used, if they invested in one system and then made the data available to the research, to the program, within the program at first, and toward the end of the program, we put it in the public domain just about a year ago, then it would be a great resource for advancing phenomics, for evaluating how different sensors work, and evaluating the sensors alongside one another. For example, being able to look at the same picture with a multispectral and an RGB camera and a hyperspectral sensor to understand what information you can actually get out of each one. In addition to that, not only did we make the data open, but all of the software is open, and it's been used. It's continuing to be used for processing the data on the gantry. It's been refactored and is now called the Fido Oracle. We also use it for drone data processing, a descendant of the software that we developed for this project. And finally, there's open computing. So there's open computing interfaces that are provided by the National Center for Supercomputing Applications, and there's also computing provided by Cyverse, where people can come in and make use of the data. So to you, it's low cost. That's the idea, is that it's ultimately basically free. You don't have to do the work to generate the data, just to understand it and analyze it. So here's a picture of that box that was hanging down from the scanner. It's got a pair of stereo RGB cameras. It has a thermal camera, the FLIR. It has a 3D laser scanner, a hyperspectral, and then this PS2 camera. It has a flash of light, and then it looks at the response of the plant's photosystem over time. So it actually is a video that's about a second long with images, about 100 images taken in sequence, so you can see the response of the plant's photosynthetic machinery to the flash of light and understand its physiology in that way. So now I'm going to show you some examples of the data that we collect. These are all zoomed in so that you can get an idea of what the actual resolution is. So the pixelation that you see is at about 1 millimeter resolution. I think it's a little bit better on the RGB camera, a little bit less than that on the FLIR camera. One of the key algorithms we have is a plant mask. So we have not only the images, but we provide the plant masks as a data product. And one of the challenges that we had, we imagined we'd be able to put together images in the same way that you can with a drone, to stitch them together into a field. And this is an approximation of that. The problem is that when you have a camera that's only 2 meters above a 3 meter tall plant, the geometry would makes it much more of a challenge than when you're in a drone. And so you get a lot of distortion. It's not only do you get the distortion at that scale, but when you're looking at the plants at such high resolution, you'll get a geometric distortion if you try and stitch it. So the full field mosaics are tiled rather than stitched. And this is useful for a lot of things: quality assurance, also for estimating canopy cover, for looking at the plants. But there are cuts, like lines where the images are put together. Most of the computer scientists and computer vision people that we talk to, they prefer to work with the original images. So each plot, depending on the sensor, may have, in the case of the RGB, 20 or more images per day, taking different parts of the plot. Okay, so here's an image from our thermal infrared sensor. You can see the soil is much warmer than the plants. One thing to note about this sensor is that when you get the data, if you open it up in your image viewer, it's going to be black. But the reason that it's going to be black is because it's, like with most of the images that we've taken, we've designed it to work on to capture the data best. So we capture a high dynamic range, but when it translates into an image, the numbers tell you what the temperature of the plant is, not what the it doesn't create a pretty image like this. So this is a separate way of looking at the data. The data product will be a raster file with temperature values in it. This is our laser 3D scanner. So this is not a LiDAR, actually. It's a line scanner. And on this first image, what you're looking at is the depth, is the distance from the laser to the leaf or to the plant. And then there's an algorithm that takes that information. So that's actually a raster file. And also for a lot of analysis, this is a easier format to work in. Although it doesn't necessarily tell you the plant height itself, it tells you the distance from the scanner. It's still very useful, especially for identifying parts of the plant, like in this case, the panicle. But we've also have an algorithm that converts that into a 3-dimensional file, a point cloud, as you might be familiar with from LiDAR, but at a very high resolution. Now, as I said earlier, about a millimeter resolution. And so we have those available as both LAS files and PLY files. Those are two different, ways of looking at the same data. And we have two hyperspectral cameras. We've got one in the visible and near infrared, and one in the shortwave infrared in the longer wavelengths of V and IR. This one is the one that has finer than 1 nanometer resolution. And so in total, we have about 2,000 bands between 300 and maybe 350 and 2,000 nanometers of wavelength. And this right here, so just so also, again, so you can see the resolution of this camera, we have, the distance in meters. So each of these is about 5 centimeters, each block. On the x-axis is 5 centimeters, and the y-axis you can see a 10 centimeter distance. And so this is what it looks like at one of the wavelengths, at one of the green wavelengths. This is the PS2 camera that I was telling you about, and some of the metrics that you can measure from it. Looking at the time series over a pixel, for example, you get something that looks like this on the right, the response of the photosystem to the flash of light. So there's a lot of ways in which you can get TERRA REF data. One of the things that we did, and one of the reasons for this, is that rather than trying to create one whole platform for the data, we're using a lot of existing utilities. And so we have our a number of different databases, but it's easiest to get the public domain data that we've put either from Globus or Dryad. So Dryad has everything except for the sensor data, and Globus has all of the sensor and genomics data because it's larger and it's easier to transfer from Globus. And it would be impractical to download files like Dryad does. Kristina is going to walk through the TraitViz web app, which is a way of visualizing a lot of the phenotypes. We've got documentation. This Globus interface is where all of our public data is. And then the tutorials, Kristina is going to walk through some of those. And I just want to note that we have public domain data, and we have a lot of data that's also not currently in the public domain and hasn't been published, and it doesn't have a DOI, but it is available for use. The key difference is that what's in the public domain has gone through quality assurance and quality control. We've looked at it, we've described it, and put it out and organized it in this, in these different, on Dryad and on Globus. But the rest of the data is also pretty well organized, and it's all available. It's just a matter of asking for a permission to get access within Globus or within our trait database to access that data. So for the data that we released last year were 2 of the seasons, and this gives an idea of the frequency with which we collected data and at which the data is available. So you can see some of the sensors we run as much as possible, like the RGB camera and the laser scanner, some like the PS2, it's very specific and targeted. So that's a system that we have to use at night, and we have a particular protocol for moving it around the field, moving the system around the field so that it's not capturing the neighboring plots, next to each other. And we also have, in addition to all of the thermal, all of these sensors, we also have an environmental logger, a weather station on the system itself, and we've got a weather station. There's the Arizona Meteorological Mesonet has a weather station nearby, and we provide that data as well. So here's an example of the trait data. So a similar plot on the right for the frequency at which all of the trait data is available for season 4 and season 6. As you can see, in season 4, we did a lot of data collection, very intensive sampling of a number of phenotypes, and the work has not yet been done to compare, to see whether or not we can estimate these manual measurements based using sensor data. Now all of the manual measurements are in black, and so that's an open problem. There's so much more work that can be done. Our job in this project was really to establish this open access database, and so now the fun part begins. On the left is a TraitViz web app that we put together where you can navigate through the seasons and look at what all of these look like time series. So here's a time series of canopy cover for season 4 that Kristina is going to show how to get access to later today. And finally, here's the environmental data. So as you can see there, these are all the weather data that was collected at the site, and you can see there's some gaps. That this is common for most weather data. A lot of meteorologists will gap fill their weather station data. We haven't done that because we have a nearby weather station if you need the continuous time series. And this will tell you the information about what's happening closer to the site, mounted on that machine. And then we've got a huge amount of genomics data that's also available. So it's been sequenced, and the sequence and the SNPs are provided both on Dryad and on Cyverse. And this came out of a population, so both of these seasons came from the Biomass Association panel that was described by Breton et al. In 2016. And finally, there's a lot of other data. There's agronomic metadata. We show here the timeline of the activities in the field. We have maps of the soil. That's in the process of being published, but we studied the soil very closely. That was led by Marcus Tuller. We've got the weather data, all those hand measurements. the sensor-derived traits and the genomics data. I think Kristina is going to show going to work through the TraitViz web app, and so I will just quickly step through these. You can look at the plot level stuff. You can go over to the map part to look at the to overlay heat maps of the traits, and there's some information on how you can access the data other than getting it on from Dryad. These are the tutorials. So the quick start that Kristina is going to start working through today. It provides a lot more information about the system. And with that, I don't want to delay any further, and I will hand it over to Kristina.
Awesome. Let's make a point here. Wow, that's great. A lot of data information. So there is no application of analyzing data, right?
Oh, I'm sorry, I didn't hear you. You were blurred.
Yeah, I just said that there is a lot of data, and if you wanna, if you are over there and you say, "Oh, I can't start because I don't have data," that's not the excuse anymore. You guys have a lot of data to share, right?
Yes, exactly. Yes, and
Amazing.
contact me if you want to use it, if you want to make use of it. It is big and difficult to wrap your head around, but we're happy to help. So.
Yeah, awesome. And also, anyone can send message to you guys to have more information, right?
Yeah.
Okay, perfect. Just to let you know, because we are using internet and many devices, so if someone lost the connection, please stay there. We're gonna do our best to come back in time. So keep with us, okay? Kristina, it's all yours.
Okay, thank you. And thank you, David, for going over all of the data at a broad scale. So now we're going to start digging down into some of it. Let me share my screen.
Yeah, let me know if you need anything.
You can see that, right? Yes. Okay, so as David had talked about a little bit, we have this web app that we have for visualizing the data. And I would recommend anyone who's watching this, please don't go to it right now, because if a bunch of people try to visit the website at once, it will crash. But afterwards, definitely go ahead and look at this and play around with it yourself. But if people could sort of hold off for right now, that'd be great. Okay, so we have this web app. It's based on Shiny that is used for displaying some of the data that we have available, sort of a way of exploring like what we have. And so you can see up here, we have tabs for some of the different seasons of data. We're focusing a lot here in seasons 4 and 6, because those are the data that have gone through most of the quality control and such. And I think David mentioned that. So for example, we can look at the season 6 data. And then, so down here, we have some options in these drop-down menus. So for example, you can look at some of the different variables that were collected. And so some of this came from sensor data, and some of this was hand collected data. And so we can just look at canopy cover here. And then the second dropdown is for cultivar. So for example, so we have all these different cultivars here that there's a bunch of the genomic information for. And so we can just select one of these. Let's see here. I think this one might be the one I was thinking of. And so, yeah, so down below you can see there are these 3 tabs here. So the first one is a plot that shows canopy. So canopy cover is the % of the ground that is covered by plant material. And so you can see this is a time series of canopy cover for this cultivar. And there are actually, if you look at, so the points are for the data, and then there's these lines connecting them. And you can see there are 2 lines, probably because this cultivar was planted in 2 plots of the field. And so we'll actually talk about the field, how it's set up too. So if you click on this map button, hopefully this will load, or map tab, I suppose. I'm gonna try to run cultivar.
Yeah, this Shiny application is really great for showing data, right?
Yeah, this is really helpful, and it helps give a broad overview when it is working.
Yeah, don't worry, don't worry. We know that's alive.
Yeah,
that's
this
alive.
is like live demos of anything can always go a little wonky.
Yeah,
well, okay, so when this is working,
seems that David is talking something here.
Oh, I was going to say, I believe that if you have, if you go to the cultivars field and you make it so that you're not selecting a cultivar, you'll see all of the data at once.
Okay.
Yeah, go to now that should show the field. And then.
Okay, sweet. That's what I was trying to do.
Awesome. Very great.
Okay.
Very great.
You're right. I forgot about that. Okay, so yeah, to be able to look at all of the cultivars, that makes sense because I had selected single cultivars, and so you could see like little green boxes that were subsets of this. But so yeah, so this is the Google Maps. It's a Google Maps interface. And so this is what the field looks like from top down. And so you can see the field is long and skinny. Can't remember how long it is exactly. But within that, we have all these boxes. So these each are the different plots that the field is split up into. And so for the season 4 and 6, different cultivars of sorghum were grown in different plots. And then they also had some additional treatments related to water and such. But this is pretty nice. You can hover over any of these and it will show you what cultivar was grown there for that season, and then also so each of the plots, how they're identified. And so for this system, the plots are identified by range and then by column. So range is basically row, and then you have column, and then they're numbered. And you can see the actual data too. So for this particular plot, the canopy cover at that time was 39%. So yeah, this is a pretty neat app that is useful for, okay, that's right. You can also change what time it is. I was thinking there was a way to do that. I kind of forgot. So you can change what date of the collection that you're looking at too by changing this. Yeah. So now so what I was originally looking at was later in the year, and a lot of the plots were lighter in color, lighter green, because there was less canopy cover. But this is a little early in the year, and so now it's darker green because you can see a lot of these values. That might be too small for you to see, but the values are in like the 80% or 90% because this is the thick of the growing season. So yeah, I think that about covers everything. And then I think this download was the information. Oh, yes, that's right. And then this download tab right here in the middle showed what David had showed in the presentation for alternative ways to get the data, not including the Dryad data that we're going to look at here in a bit. So yeah, that is the web app that you can use for sort of interactively exploring the data and looking at it. Okay. Oh, the field is 20 meters by 200 meters. Sorry, I didn't know that off the top of my head. Oh yeah, and you can, that's right. There's a button that you can click here. And I can't remember what these show. I think if you unselect this, it shows, oh yeah, so this shows the actual like images that were taken. So these are those camera, I think these are the RGB images. And so David had showed in the presentation where like they basically stitched or put all of those tiles together. And I think this is the same thing. So yeah, you can either look at, yeah, so this is sort of the summarized data when you click on the heat map, and so when you unclick the heat map, this sort of shows the actual underlying data images. Okay, yeah, thank you, David. Did you want to say something about that? I think I can't hear you. Are you muted?
Yeah, I was. I'm sorry. I wanted to point out that this is a very low resolution version of the full field stitched image, just because each entire full field stitched image, I can't recall, but is like 50 or 100 gigabytes or so, and this is very small. It still takes a while to load, but it allows you to look, for example, at the canopy cover values that we've got and what actually happened underneath. So it can, and then you can find this particular image on Globus. One thing I wanted to mention is that these things are called like the name of this is stereo vis IR sensors, partial plot, sorghum, sun, FLIR, ESIG, etc. These are those are the names of the scans, not the names. Those are and the sensors that were targeted during that scan. But I know at least this one is an RGB image as opposed to a FLIR image. So that's all I wanted to point out. But
does that vary? That varies for the different seasons?
I think mostly like what we have here, what you can see is RGB images, but we do have stitched images for the FLIR as well.
Oh, they're just not in the web app. Yeah, you can't see them here. Okay, that makes sense. Yeah, that's the two different sensors for the scan 1 and 2 here. Okay. Cool. Thank you, David. Okay, so yes, so this web app is a good place to start if you're interested in exploring the TERRA REF data. And we do want to continue making improvements to this and add more data and such as time goes on. One of the other entry points for data that David had mentioned was the Dryad data download. And hopefully we have instructions or installation instructions for actually installing that from Dryad. And I guess I should pull up the actual link for the Dryad data. Hopefully my screen isn't working. There we go. So yeah, and one of the main sources now for the data for seasons 4 and 6 are hosted on Dryad, the data repository. And so this is the landing page for that. And so there's a little bit of information on here about the data set, some of the stuff that we're going over now. And then if you want to download the entire data set, which I already did, you can click this button here, download data set. Or you can download it in a more automated way using R. And so I already downloaded it. Hopefully everyone has had time to download it. It can take a minute. It is a little bit big. But if you can download that, then you can actually follow through the rest of this tutorial. So as I said, I have already downloaded the data. And so it comes downloaded as a zip file. This one right here. Hopefully that. Well, you can't see that. Hold on. I need to change my screen sharing.
Yeah, just for let everyone know, the link for the data is here in the description, okay? So if you want to go there, you're going to find it.
Yeah. Oh, and I guess also, I don't think we noted, but we have all the notes for this entire tutorial on GitHub. Open in an open repository. So we'll post the link for that too, so that if you miss anything or want to look back at some of this stuff, there's a full set of notes for what we're going through here. That should be a good resource. Okay, so when I downloaded the Dryad data, this isn't too small, but you can see in my file finder, I have an RStudio project already set up to look through this data. So I downloaded that data from Dryad, and so there's a bunch of information in here about the data, and I'll kind of walk through what is available. I don't know if I can make this. I can't make this text any bigger, so it's a little small. Sorry about that. But so I unzipped the Dryad data download, and so I opened up this folder. And so in this folder is a bunch of data and metadata about the TERRA REF data set. And so the best place probably to start is that there's this README, readme_terraref_dryad.pdf. So if I pull that up, geez, okay. Screen sharing is being problematic. These different things. Okay, so this is the PDF for this Dryad data download. And so what is contained in here is, it's actually not a super long document, but it's got a lot of useful information, an overview of TERRA REF and what was collected and how the data can be used. And some of the information in here is also specific to the, oh, yep. You can't see that. Okay. Sorry, the screen share is messing up when I try to share my whole screen. But so if you won't be able to see on my screen, but if you open up this PDF yourself, you'll be able to see this README with all the information in it. And so some of the information in this PDF too is specific to what's contained within the Dryad download. It's got a description of what's in each of the folders and such. And then also it's got some useful plots in there of what data is available. So in David's presentation earlier, he showed the plots that were sort of time series for when sensor data was collected across the different seasons and when the trait data was hand collected versus the sensor collected data. And so if you want to look at those plots again, they're within this README. So that's a very useful place to start with the data. And then switching back to RStudio. Okay, there's also a code subfolder in here. That's not something we're going to touch here, and you don't necessarily ever have to look at. But basically what's in there is the sort of reproducible pipeline to generate everything that's in this Dryad download. So because the data is stored on a database, and then so the code folder contains sort of the path to get the data from the database Betty into this Dryad download. Again, not something that anyone necessarily ever needs to look at, but if you're interested, there's sort of that reproducible component there. There's another folder called metadata. This one has a bunch of metadata about the genomics data and the sensor data. And so it doesn't, I don't think it actually contains any genomics data, but it's got some information about the genomics data. There's also a subfolder for weather that has the weather data from the two sensors that were talked about earlier. And we'll actually come back and pull in some of this weather data and look at it. Okay, so that's the metadata folder that's in the Dryad download. And then we have the traits folder. So this actually contains a bunch of data, the trait data. And you'll see if you click on that within the traits folder, there are two folders, one for season 4 and one for season 6. And so within that, there are a bunch of CSV files. And these are different CSV files for different trait data that was collected in different ways. And so the naming convention for these files is actually the name or the season that it's relevant to or that it was collected in, the type of trait data that was collected, and then the very last word in the name of the title is how the data were collected. So either the manually collected data by hand or the sensor collected data. And so we'll pull in some of these files and work with them too in a little bit. Okay, and back in the main Dryad folder, the last folder that we haven't talked about yet is the sensors folder. And so there's metadata about the sensor data in here. There's no sensor data because, as David had mentioned, a lot of the sensor data it's very large, very high resolution. And so we wouldn't want to provide it with this Dryad data download because, I mean, it's too big, much too big probably to just download all of it. But in the sensors folder from the Dryad data, this is a helpful pointer to get started with with finding the data that you do want to use. So actually finding the sensor data on Globus, which we will walk through. So this is a good jumping off point for seeing what's available for the sensor data. And so you can see there's subfolders in here for each of the seasons for the different types of data. There's the RGB data, a couple different formats of those. There's he talked about the 3D laser data. That's sort of like LiDAR, but not LiDAR. There's information about it in here, some metadata about it. There's the PS2, that photo system capturing system. So yeah, these are all the different types of sensor data here. Okay, and so we're going to use the metadata here in the Dryad download for the sensor data to go and actually download some data from Globus. Okay, and so we're going to look at some of the, we're going to look at one RGB image, so one of these camera images of one of the plots. And so if we look at we're going to look in this RGB geotiff plots folder. And so within that, you can see that there are a bunch of these JSON files. And so if you haven't worked with JSON before, they're basically just text files, but they have a particular format that's supposed to make them more machine readable, which means that they're not necessarily super easy for humans to read all the time, but we can still get useful information from these JSON files. So I'm just going to look at the first one of these. It's too large to open. I've never gotten that before. Wonder, okay. Huh, it's bizarre. I used to be able to open it up in the RStudio interface. Let me see if I can open it up separately. Oh, I don't want to do that. Okay, it's not very convenient.
Yeah, it's okay.
Switching back and forth.
Do you know, every time when I'm gonna show something for my boss, this always happen.
Yeah.
Everything works
Definitely
perfect, and then when you need it, it stops to work. So have your time to open the script.
Yeah, I'll have to just go slowly between each of these steps and switch screens every time.
Yeah, it's okay.
It's not the end of the world.
Awesome.
Okay, so yes. Okay, so this is the one of that JSON file. I had to open it up in a text editor instead of viewing it in RStudio. It's easier to look at it in RStudio, but that's okay. So there's a bunch of information in here about each of the images that are associated with this site and with this date. And so it's a bunch of different images in here for, I think each plot, maybe it gets multiple images per plot. So there's a bunch of these. So there's a lot of stuff here, but the thing that we're interested in right now is, so if we're interested in this, we'll say we're interested in this first image, there's this path right here. So each of these files has a path. And this path is what's going to help us find this image in Globus. And so I won't be able to leave this up on the screen because I'm gonna have to switch over, but that's okay. So yeah, we're basically gonna use this metadata about the sensor data to go to Globus and actually get, so we wanna get this, you can see the last thing in this path is a .tiff file. So that's the geotiff that we'll say we're interested in getting. So I'm gonna switch back to my browser. Make sure I get the right one. Okay, I think I'm sharing it. Okay, thank you. Okay, so we're gonna actually go to Globus. And hopefully, I mean, it's good if you can follow along, but if not, this is just something that you can do later too, because you need a Globus account in order to do this. So it was in the installation instructions to get a Globus account, which is free. And so basically how Globus is generally set up is you can transfer data between different endpoints. And Globus itself is a place to store very large data. And so we have an endpoint on Globus for TERRA REF data. We actually have a couple of endpoints. We'll be using the public one. And then you can also use Globus to send up an endpoint on your computer. And so basically what you can do then with Globus is transfer data between different endpoints. So I'm going to show you how to transfer data from the public TERRA REF endpoint to my local computer endpoint, which when you set up your account with Globus, it should automatically put you through the process of setting up your computer as a Globus endpoint. Oops, I'm gonna have to log in. Should already be logged in. Okay, yes, so this is what Globus looks like when you're logged in. And so the first thing that we're gonna start with is looking at the endpoints. And so, sorry, this is a little small on the screen, but on the left-hand side menu, there's an endpoints button that you can go to. And so I created my own, if you go to the administered by you, it should show your endpoint. Oh, and mine is showing red. And that's because you need to, in order for this to work on your local machine, to set up your endpoint properly and have it run, you also have to have Globus Connect running. So let me go and start Globus Connect. Should take a moment. And all of this, the instructions on Globus when you sign up, it should walk you through all of these things. Okay, so now I have Globus Connect running. Okay, and now my endpoint, which you get to name, I just named it my MacBook, I can see that it's running because this is green instead of red now. Okay, so this is the endpoint we're going to transfer data to. The second thing you need to do is get access to the endpoint that you want to transfer data from, so the TERRA REF endpoint. And so you can do that by going up to the search bar, typing in NCSA pound or hashtag, I guess, depends on how old you are. Then type in terra-public. And so when you search for that, then the endpoint for the public endpoint for the TERRA REF data should show up and you should be able to access it. So yeah. So now I have access to both of these endpoints. And if you're following along, maybe you do too, or maybe you will in the near future. Okay, so now we have the endpoint set up. Now we need to actually transfer data from the TERRA REF endpoint to the local endpoint. And so to do that, we go up here to where we started on Globus. There's this file manager. And so basically, the left side will be specifying the local endpoint, and then the right side will be the TERRA REF endpoint. We'll set them up. So I'll walk you through this. Okay. So on the left side, I'm going to select my local endpoint, which is called Kristina MacBook Pro. And then I'm going to go and find where on my, so this right here, what should show up if everything is set up correctly, it should show you your local file system on your computer. So if you're in our browser, you can actually see your local file system. And so I'm going to navigate within this to my desktop. And then to a folder called PhenomeForce, which is where I set up my RStudio project. And so this is where I want to transfer the censored file from Globus onto my local machine. Okay, so now we have everything set up for the local endpoint, and now we need to set it up for the TERRA REF endpoint. So on the right-hand side in the collection, I select the public TERRA REF endpoint. Okay, and now we can see the file. This is now the file system for this TERRA REF endpoint. This is all the files that are available through this Globus endpoint. And so I'm going to use that file path that I had before. I'll actually pull up to be able to look at it. I'm going to use that file path as a guide to get me to this .tiff file that I want to download. And so I know that I'm going to need to go to season 4. And then we want to look at the level 1 plots folder. And then we are looking at the RGB images, RGB geotiff. Okay, now these are sorted according to date. Sorting according on Globus, it's date and then the plot, I think. We'll see here in a second. So I'm going to select the first date, March 26, 2017. Yeah, and this is. Now this is too small for me to be able to see the full name of the folder. I wonder if I click on it. Nope. Okay, well, so I wanted to go to the, so these now are arranged by plot, and I wanted to go to the plot that's range 21, column 16. I can't see the full file name here, though. I don't know how to make this different sizes. Okay, hold on, let me. Okay, well, it probably doesn't matter too much. I'll just pick one of these at random. So within each of these, within each of the plot folders, there are a set of geotiffs. And so these are all of the camera images, the RGB images in particular, that were taken on that date for that plot. And so you can see there's a handful of them. And so I'm just going to download the first one. Start going a little off script here, but I think it'll be fine. So once you select the file that you want to download, you can just click the Start button, and it should transfer it over to your local endpoint. See if there's a way to click on View. click size.
You can also click on the last modified one under view. Yeah.
Okay, David's trying to help me figure out how to do the resizing. I don't know. Usually you can see the whole thing. Okay, that's okay though. Yeah, did you want to say anything about that?
No, not. You're doing great. I just was put in a comment in the chat that that's how you could get more space on the screen. Well, I guess the one other thing is that up above that start button, you can copy and paste that string from the JSON into there. And I'll also point out that the whole idea is that if you were going to be processing a lot, you could automate this entire process, telling through Globus's API, telling it each file that you want to get after you've discovered which ones you want, or you can download it in bulk, we're doing today.
Yeah, I guess that makes sense. Yeah, this is very much like a GUI, like a point and click interface right now, the way that we're using it. But yeah, then once you figure out which files you want, you can use the API to pull down larger numbers of files at one time in a more automated way, which I haven't actually done myself, but I think would probably be fun to do. Okay, so that file transfer is probably complete. You can actually see it had this little green pop-up here where you can look at the details of the transfer. And it says it succeeded, and it took 50 seconds. So now if I look on my local computer, that file should be there. And it is. Oh, I'm gonna have to switch my screen back to RStudio now. Okay, so that's how to go onto Globus, and we found one particular file that would have been of interest to us. And that's sort of the approach that you can take to download data from Globus. And Globus themselves, they have like tutorials and stuff and instructions online that are helpful too. So, okay, so now that we have downloaded that file, you can actually see I downloaded it to our project that I'm working in called PhenomeForce. I just did that so that it would be easier to deal with file path issues and stuff. So now that we have that .tiff here with its long descriptive name about what it is, we can go ahead and we can read it into R. So I'm just going to open up a new R script. And so R has a pretty good set of packages for working with spatial data. And one of them is raster, if you haven't worked a lot with spatial data in R. So this is one that's useful for gridded data like this. So I'm going to read in or load in the raster library, and then I'm going to use the raster function from that to read in this .tiff file. Oops. Okay, it's got a long name, so I'm going to autocomplete. Okay, so we read in that .tiff file. And so you can look at the format of the file. You can explore it in different ways using R's tools. We're just going to go ahead, for the sake of brevity, we're just going to go ahead and plot this to see what it looks like. And see if it looks like what we expect it to look like. And it'll take a minute because these files are a little bit big. But once, okay. Okay, so this is a different one than I'm used to. I had a different TIFF file set up as an example, but you can see down in the lower left-hand corner. So this, I think, is the entirety of the plot that this image was taken in. And so this image just took this little, here, I'll make it a little bigger. So this particular .tiff image for this plot only covered this small bit of the plot in the lower left-hand corner. And so you can see, because this is a little early in the year, I think this was April. Yeah, this was April, that the canopy cover here is only about 30 to 40%. So because the plants are still growing, it's still kind of early. So yeah, based on this, you can kind of get an idea for the sort of things that you can do with this sensor data. Once you've downloaded it, you can go ahead and use all these different tools to plot lots of the different files all at the same time, or combine them, put them together, run different analyses on them, kind of whatever you want, depending on whatever question you're interested in. So yeah, so that is the sensor data. That is a sort of an overview of what sensor data is available and how to get it. Okay. So the next thing that we're going to look at a little bit is the trait data that's available from the TERRA REF data set. So as I said before, oops. So we can go back to the Dryad data download now, because the Dryad data has all this trait data in it. So it's a little bit easier to access than sensor data because it's not as big. And so if you go back to this traits folder, we're going to go ahead and look at one of the trait files for season 4. And so, as I said before, you can identify what trait is in a particular CSV based on the name. And so we're going to go ahead and we're going to read in canopy cover. So there's these canopy cover files here, and we're going to look at the sensor derived data, not the manual derived data. I'm just going to read that in. It's a CSV, so it's a pretty easy process. So I'm just doing the file path for that. And then we want season 4 canopy cover. Okay. And so that reads in quickly because it's just a text file. And so if we open that up, this is an example of what, so all the trait files are sort of organized in the same way. We have it so that each row is for a particular date, location, cultivar, and then trait. And so you can see all that information here. There's information about the plot that this data, each of the data points comes from, the species name, cultivar, treatment if there was one. Yeah, date. So the trait here, all of these should be canopy cover because this is in the canopy cover CSV. And there's a little bit more information. Oh, and the most important thing is the mean column. That's where the value for that measurement is. So this is mean here is canopy cover. If you open up a different trait CSV, it'll be the value for whatever that trait was measured. Okay, so we're going to go ahead and we're going to plot some of this canopy cover data just to look at it and see what it looks like. And so in order to do that, because I'm going to plot a time series of the canopy cover data, I'm real quick going to go ahead and have to change this date column into a date format to make the plotting easier. So I'm going to use dplyr for that, which is a pretty basic, important R package for, I guess not basic, but a pretty commonly used R package for data cleaning and exploration. So I'm just going to recreate that. Okay, so basically what I just did here is I'm going to rewrite over the data frame canopy cover, but changing that date column to be a date type. So basically everything stays the same besides that. Okay, and then we can just go ahead and plot this. I'm going to plot it using ggplot, which is the R package for plotting that I'm most familiar with. So I'm going to plot that canopy cover data so we can actually look at it and see if it looks like what we expect it to be. And again, I'm plotting the time series of the canopy cover for across all the plots for this. This is across all of the plots for and all of the dates for season 4. Okay, and so you can see there's kind of a lot of data here. And this is sort of what we'd expect to see from canopy cover measurements throughout the course of the year. You can see this sort of like green up. The plants are really growing pretty quickly earlier in the summer, and then they all sort of reach a sort of maximum plateau canopy cover value below 100, but some of them get kind of close. And then later on in the season, that starts to vary more as the plants start to senesce and such. So yeah, that's just an example of how to pull in some of this canopy cover data and look at it. We also, you can look at this just by, you can filter by lots of different things based on what parts of this you're interested in. But we can say, like, if you're interested in looking at a particular cultivar, we can just go ahead and plot the data for one particular cultivar. And so I'm going to just look at the data for this one kind of random cultivar that I chose, but based on what you're interested in, you might be interested in certain cultivars. So this is PI 656026. And so I'm going to create a new data frame that just has the data for that cultivar in it. Okay, so now we have a much smaller data frame that just has all of the data for this particular cultivar. And so I can plot this just like I did before for the entire data set. And I know already that this cultivar was actually grown in a couple of plots. So this is the sort of thing that you wouldn't know until you start exploring the data. But I know that this cultivar is grown in more than one plot. So I'm going to actually color the points in the plot, or color the points in the graph plot by the field plot. This is taking up, probably hard to see because my text is really big. I'll try to fix this. Yeah. Okay, so yep, this is a time series now just for this one particular cultivar, and then you can see that this was grown in this season for 2 plots. So the one at row 20, column 3, and another at row 50, column 10. So yeah, that is sort of just a quick look at an example of some of the trait data that's available. There's a bunch of other trait information that are in that Dryad data download. You can see there's lots of things that could be of different interest to folks, depending on what sort of like plant physiology that you're interested in. Okay. Okay, so if seems like there's no questions or anything, so that's good. Hello.
Hi, sorry, I'm unmuting myself. Can you hear me now? My mic wasn't working.
Yeah, I can.
Your data looks really nice. I like you seem to have very tight temporal coverage in your seasonal plots. Could you explain like what you might do with like canopy cover temporal data in your program in particular? Is there anything you do with that? Do you fit any models or anything like that?
Well, we actually are going to. So once I've gone over the weather data, I was actually going to show an example of how to sort of look at how temperature could affect canopy cover.
Very cool.
I don't.
It's right up my alley.
So, okay, good. But yeah, I mean, imagine there's a lot of different things that you could do with this, especially with having so many different cultivars grown in at different spots in the field. And this data, I think in particular, there was a drought treatment applied to it. So sort of looking at the impact of that on plant growth. Like I feel like there's a lot of questions that you could explore there.
Absolutely. I'll leave you to get back to it.
Okay, thank you. That was a nice, I mean, it's a nice like view to the future. Maybe I should have mentioned that. Yeah, so for the rest of this, I was going to go through pulling in some of the weather data. And then I was going to use the weather data and then model sort of how that might be affecting plant or canopy cover. So as an example of what to do here. So yeah, thank you, Claire. Okay, so we'll first look at some of the weather data. Again, all this like sort of prep work has to be done. You have to understand the data set. You have to, or the many parts of each of these types of data sets. It takes a while to understand what all is available. What you can use together. Sort of looking at things like the temporal and spatial resolution is something that, like, as someone who's worked with other people's data a lot, it can take a lot of time and effort to sort of parse through all of that. And what we're really trying to do here with TERRA REF is make it easier, like lower, especially for data that's so sort of, it has such a high resolution. Like we're trying to lower the barrier for people to be able to understand the data to then be able to use it. And so this is part of the what some people see is sort of like the barrier to them getting to the actual answers to their scientific questions. But it is a huge part of the process. And it can be very hard to provide this data in a way that is easy for people to use. And of course, we're not perfect. No one's perfect at doing this. But yeah, we really appreciate feedback on how to make this more usable for scientists and researchers. So with that said, I'll get off my little soapbox and I'll read in some weather data. So in our Dryad download, the weather data was in the metadata subfolder. So we have this weather data. And as David mentioned, there's two sources of weather data or meteorological data for this site. One of them, which we'll look at the AZ Met one, and that's from a sort of more general Arizona network of meteorological sensors. And then there are also the sensors that were right on the gantry. I think they were on the gantry itself, but it was at the field. We're going to use the AZ Met data because it's a little simpler. And so that's in this folder right here. And you'll see that we have data, weather data that's available at hourly and daily resolutions. And then we have data for the 2 seasons. So there's 2017 and 2018. And so to make sure that we're getting the right weather data to match with this canopy cover data, because we're going to bring them together and model them, I'm going to go ahead and just look and see what the date range is in the canopy cover data to make sure I get the right one. Okay, and so yeah, the canopy cover data was from season 4, which was collected in 2017. So we're going to use one of these 2017 AZ Met files. So I'm going to read, and we're going to use the hourly data. And so for right now, we could just use the daily data, but a later step is going to require us to use the hourly weather data. So that's the one that I'm going to read in. I'm reading this weather data, and we'll call it weather. Just gonna have to type in, get this long file path. Well, it's not too long, could be worse. Okay, so I have the file path to the AZ Met folder, and then I want to read in AZ Met 2017 hourly. Okay, so once that's read in, we can go ahead and look at it and see the structure of this weather data. Okay, so you can see stuff here that looks like it's probably wet. This is probably a year. There's some measurements here. But this weather data doesn't have very good column names. And so the way that you can figure out for this weather data, what the columns are is there's a README in that folder called AZ Met _README. This is a little, okay. So yeah, there's a bunch of information here that's about this weather sensor. But what we're interested in is column names. So if you scroll down, right here, we have descriptions of the hourly files. So we're looking at hourly file. And so this right here is a list of, so for each column, what is in that column. And so the ones that we're interested in here for what we're about to do is, well, we want to get the date. So we're going to use the first 2 columns, the year and the day of year. And then we're going to be looking at temperature. So air temperature is the 4th column in this data frame. There's also other useful information in here, including, I think, the units. Yeah, they have the units in here for each of these too. So yeah, this data set needs to sort of be put together a little bit more before it becomes usable. So we're going to go ahead and make this usable. So what I'm going to do here, I'm kind of going to do it all in one shebang using dplyr, is I'm going to get a date, because there's no date column in here right now. There's just year and day of year, and I want a date column. So I'm going to generate a date column from this, and then I'm also going to calculate the mean daily temperature, because right now each of these rows is hourly. So I'm just going to get the mean daily temperature, and we can plot it and make sure it looks okay. So I'm going to create a new data frame that will have all that stuff in it. Oops. So first thing is creating that date column. And like I said, I'm not even using the year column here. I'm just using the day of year column. We'll talk about this in a second real quick. So yeah, so this date column, basically what I'm doing is I'm saying if we say the origin, so the start date was the last year of 2016. And then it's going to iterate using the day of year column, so 1, 2, 3, 4, 5, to figure out what the data is. So the day of year 1 will be the first year of 2017. Okay, and then I only am going to end up wanting that newly created date column, and then I want the temperature column, which was the 4th one, which has this not particularly useful name, x13.3. So I'm just going to pull out those 2 columns. Sorry, it gets a little crowded here with having the text the right size to be able to see on the screen. Okay, and I'm going to rename the temperature column to what it actually is, so it's easier to work with. Okay, and then I'm going to calculate the mean daily temperature. Okay, and I'm going a little quickly over the R stuff here because that's not really the focus of this. Like, there's many ways, obviously, that you could approach this data and sort of slice and dice it and inspect it. So, and you can always go back and look at the code for this later on. If you have questions about it, feel free to ask me. I love talking about R. So, okay, so now we have this new data frame, which has mean daily temperature for each year of 2017 in it. We're just going to quickly plot that because it's good practice to look at data to make sure it looks like it should. So an easy way to identify errors. Errors either in the data itself or errors with how you sort of have processed it or approached it. So I'm just going to create a little scatter plot here of that. Okay, so yep, we have mean daily temperature across the course of the year. Gets warmer, gets colder. The values are about what we would expect for Celsius in Arizona. So yeah. So that is the weather data, and that's this is the this was the hourly data instead of the daily data. But then we summarized the temperature by mean day. We also could have just plotted all of the temperature data hourly. It just would have been something probably pretty similar, but with a little bit more variation and a lot more points. So,
I'm back with another question for you. So
yeah.
Is your temperature data, is that just like a weather station beside the field, or is that actually in the TERRA REF's big car-sized box that it collects temperature data?
So this data that we're looking at is from just sort of a general sensor that's nearby. Because the field is in sort of this bigger experimental station run by the University of Arizona. And so I actually don't know how close it is to the field, but I mean, it's like not very far. But then, yeah, I think, and David might actually know, he knows a little bit more of the details of this than I do, but yes, there's a sensor that's, I think it's on the gantry, right?
Yeah, so there's a combination. There's some sensors, most of the sensors on the top of the gantry system, and there were a few that were on the box itself. Okay. And I apologize, I can't recall which ones were on the box itself. Yeah.
I was just interested to know, like, because obviously the overall temperature will be fairly similar, but when you're comparing things for like infrared, if you wanted to know if there are fluctuations, it's important that you're close for that. So cool, thank you.
Mm-hmm.
Awesome. Yeah, that's a good question. Oh, okay. Yeah, there's a lot. I mean, because so many different data types were collected and lots of different sensors and equipment and stuff like that. Like, there's a lot that can be done, but also can make it a little overwhelming to figure out.
So we have someone in the chat that got a bit of an error. Just take a quick look and see if you've seen that before.
I'm assuming that was an error from the raster function. I have not seen that before.
All right. Well, we'll just keep going from now. Maybe we'll see if we can figure it out in the chat.
Yeah, definitely. I mean, of course, one of helpful suggestion is to Google the error message and see what happens. It could just be like a problem with package versions or something like that, possibly. So yeah. Okay, so the last sort of bit of this that we're going to do is we're going to combine the canopy cover data with the weather data, and we're going to model the relationship between them. And so the first step of this is that we're going to take the temperature data from the AZMet, and we're going to calculate growing degree days. And so basically, I mean, some of you might be much more plant folks than I am and know this better than I do, but basically growing degree days is a metric of how much heat plants have been exposed to throughout the course of the year. And so as they get exposed to increasingly more heat, it can cause phenological stages to occur, things like flowering, dates of flowering and such. So we are going to calculate growing degree days. And to do this, we're going to go back to the original weather data set with the hourly data, because what we need from the weather data to calculate growing degree days is the mean of the lowest temperature of the day and the highest temperature of the day. So we can't just take the mean of all of the data for that day, because we specifically want like sort of the lowest and highest values. Okay, so I'm gonna have to go back and repeat some of the steps to sort of clean up the weather data, but then do this like last different step. So I'm gonna create this new data frame. Wow, okay. And so just like before, I'm going to have to get a date column. This is exactly the same as before. Actually, I could probably just copy and paste some of this, might be easier. Yeah, I'll do that. So I'm going to type it all out by hand. Okay, so I'm just copying the first 4 lines that we used before for sort of cleaning up this data, so that we end up with 2 columns, one that has the date and one that has this the hourly temperature in it. And then so what we want to do is we want to pull out for each of the days, we want to pull out the lowest temperature and the highest temperature and then take the mean of them. So I'll just do that real quick. Oops. New line so you can see it. Okay, and so what we hopefully have out of here then is going to be for each day, the minimum temperature and the maximum temperature from those hourly values, and then the average of those. And so if I did it right, and it doesn't seem like there's any errors, that looks like that's what we got. So we have this column now of average temperatures based on the lowest and highest temperatures of the day. Okay, and so with that now we're going to get, so this isn't GDD yet, this is just setting up the temperature data for it. And so growing degree days is a measure of the accumulation of heat over time that a plant is exposed to, but specifically only over a certain base temperature, because I guess, I mean, below, you know, a certain temperature, the amount of heat to a plant is going to be negligible. And so we're going to create this base temperature. We're going to choose 10 degrees. So basically anything that's below 10, the plants, we're assuming the plants don't really care about, but any temperature that's above 10, it does matter. So to get our growing degree days from that, wow, oh goodness. So what I'm going to do is I'm going to create a new column for the actual growing degree days, where it's going to look at that average temperature that we just calculated. If the average temperature is over 10, it's going to return the value that is the average temperature minus 10. Otherwise, it's going to return a 0. So I'm using an if-else statement for that. This is going to be way too long for this line. So wow. Oh my gosh. I know how to type, I promise. Okay. And so I'll did something wrong. I'm missing something here, aren't I?
I guess it's only the sign, maybe. Man, you're being brave to type the whole code. Like I'm not able to do that.
Yeah, I've had a lot of practice with live coding, and I'm used to making errors regular. I mean, this is like what it is to work on it really, every day.
Yeah, that's amazing. I have my script and then I just keep copy and pasting, otherwise I can't do it.
Yeah, well, I can try and copy and paste and see if what I have in my notes is going to work better. Bugs me though. I always want to figure out where the error came from. Oh, I know what it was. I think I didn't actually run the base temp line, so it didn't have a base temp object yet.
Here we go.
It was like, why are you trying to do something with an object that doesn't exist?
Here we go.
Okay. Oh, it looks like David caught on to it. I'm sorry, I didn't see your message. David caught the error before I did. Okay, so let's actually look at that and see if it did what we expected it to. So what we want to return in this GDD column is a 0 for values under 10. And then for values that are over 10, we want the temperature, average temperature minus 10. Basically that those increments of heat above 10 are the ones that matter. And so it looks like that worked just fine. So yeah, some of these rows in the growing degree days are 0 and some are not. Okay, so we are still not quite there yet because we want here's the cumulative heat that the plants have experienced throughout the course of the year. So the last step here is to take this column and add these amounts incrementally so that you have a cumulative value instead of just the daily value. And I'm actually just going to add that right on here. Add on another column using the mutate statement. I'll call this one GDD cum, using this cum-sum value. Okay. And we don't need all these other columns now, so I'm actually just going to pull out date and then this final column with the GDD in it. Okay, so now we have by day the growing degree days throughout the course of the year cumulatively. And so like before, we can plot this. I'm going to plot this time series to see, just like always, you want to check and make sure that it kind of looks like what you expect it to look like. And if it doesn't, then investigate why not. It could be something interesting in the data. But no, we kind of have what we have here, what we expect to see, which is a sort of rapidly increasing amount of heat up through the summer, and then it starts tapering off when you get into the end of the year because it's getting cold again. Okay, so now we have growing degree days. We took our temperature, our hourly temperature data, and we calculated growing degree days as a bit of more of a useful metric for like what plants actually kind of care about. It's kind of anthropogenic. Plants don't really care about anything, but a more useful GDD is a more useful metric for like why plants would respond. And so what we're going to do is we're going to model the relationship between this growing degree days and then the canopy cover data that we already looked at before. And so my first step for doing that is to take the canopy cover data and then combine it with the growing degree days data. So I'm going to have a new data frame for that. Oops. What was it called? Canopy cover cultivar was the one. And we're just going to look at it for one of the cultivars. Sorry, I didn't mention that. But we're just going to look at it for the one cultivar that we looked at before. Okay, so what this is just doing is taking the canopy cover data, which had fewer dates in it, if you remember, because this is the data weren't collected every single day of the year. And then we're combining it with the growing degrees data that we just calculated. So we're combining it by date. And then I pulled out just the columns that we'll need from that. So now we have this data frame that has for each day, it has the mean. Remember, mean is the column with the canopy cover data. And then we have that new GDD that we just calculated. Okay, so we can look at this relationship. Oh, Oh yeah, oh, I skipped something. Okay, and we are going to look at the canopy cover data only up until the canopy cover kind of reaches this asymptoted peak before it starts dropping off. So we're actually I'm gonna subset the data, so we're only looking at the data up until July 1st. So I'll just add that on here to the end of, I'll go ahead and filter the date. Okay, so now it's a subset of that data set that we plotted here, only going up through to, or not here, sorry. We haven't plotted it yet. Let's go ahead and plot it first. We'll talk about it. Okay, so now what we have on the x-axis, it's showing the growing degree days, and then on the y-axis is the canopy cover, and it's only up through so July 1st, up through kind of where that canopy cover increases and then sort of tapers off. And so you can see there's this sort of nice, pretty nice looking relationship. And so we think that one model that sort of would be good for capturing this relationship is a logistic growth model. And so I, ahead of time, have put together that. I don't know that I could model something on the fly in front of an audience very easily. And so something very simple. But so, okay, so I have this. Oh, it's hard to see because the text is so big. So I wrote this function for modeling logistic growth. I won't go into the details too much here, but if people want to talk about the details, we can later. But basically, there's I specified a bunch of parameters based on the data here, and then modeled this relationship. And so hopefully what this does then is it can provide a good relationship for, based on the growing degree days, what would you expect the canopy cover to be? Okay, so I have this function. I need to run it so it can actually be used. And then I'm going to go ahead and I'm going to run the function on the that cover GDD data. And then I'm going to put that into a new column called predictions in the same data frame. So that cover GDD, which is where the data is coming from. Create a new column called predictions, which will be the predicted canopy cover based on growing degree days. So I have my new function that I just created that I'm going to run on my data, because you can see the function, it says data as the input. And so it's this data frame. Oh, I didn't like it. There's an error. Oh, that's right. Oh, okay. This actually needs to be called cover GDD underscore model. I forgot that I named the data frame something else. Okay, sorry about that. Go through and fix all of these. This is what happens when you change the name of a data frame right before presenting on it. Okay, so it looks like that worked now. Okay, so now we have this column of predictions from our logistic growth model. And these are predictions of canopy cover. Okay, so in this final step, I will plot those predictions as a line on this data. So I'm actually going to copy. Oh, no, I'm not going to copy it. I'm going to do it from scratch. Okay, so I'm going to plot the data on the underlying data as points, and then plot this model on top of it as a line, the predictions. So we have our data frame. First, we're going to plot the points. Should look like what we already have here. And we're plotting mean as the canopy cover here. I could have renamed that column, and that probably would have been a good idea, just to remember what it is. But I'm going to plot a line. Where the x-axis is the cumulative growing degree days, and then the y-axis is the the predicted values of canopy cover. And I'm going to make that line orange so it stands out a little bit more. We'll try to fit this all in the same view. Okay. So when you plot this, we'll see, and there we go. Yeah, so it seems like this model is actually a pretty good fit, at least for this cultivar. And so one of the next steps, and I think I have done this, or I think the tutorials actually have this in it, is to go through and try to do this for each of the cultivars in the data set. And then to compare the inflection point of this model for each of the different cultivars to see how they vary. Because that is kind of a sign of how the plants are growing in response to growing degree days. You can imagine some of the cultivars of sorghum here might, like, you know, maybe it's too much heat for them. They can't tolerate it as well, so they don't grow as well. They don't sort of fill out as much as the other plants do for some reason. So yeah, okay. And so that is just an example of one of the ways that you could use this data and go ahead and try to model relationships between some of the things that were measured here, and try to get a better understanding of what is sort of going on at the ecological or physiological level. So yeah, that was all the material that I had prepared for sort of the walkthrough component of this. But we can do, if people wanted to do some troubleshooting, or if people want to ask more questions about the data. David knows a little bit more about the full extent of the data too. So yeah. I don't know if there are other questions. I haven't checked the chat.
There's nothing new in the chat right now.
Yeah. First of all, thank you. That's amazing, man. I'm really impressed with your coding. I need to be honest. How you memorize all those lines is quite hard. It's really impressive. Yeah, thank you for sharing.
Well, I'm following notes, by the way. I'm not really making this up on the fly. That would be, yeah, I'm not that. I'm just a normal person. I can't do stuff like that, like a superhero. Yeah, I was following along with notes, but I did write all the code that is here. But it took
awesome.
more trial and error than it looks like.
If it was dancing, it was like freestyled, you know, and then that's really nice.
Thanks.
Yeah, thank you. Let's have time, give time for people send their questions here. But Claire and I can start. And how have been the partnerships for use this data? So normally people send a mail or they download the data directly, or for publications they need to cite a specific paper. So let's talk a little bit about that.
Okay, yeah, I'm happy to talk about that. So the data currently is on Dryad, and Dryad makes all of their data in the public domain, which technically, legally means that nobody has to cite. You can use whatever you want for anything. You can use our pictures of plants in an advertisement or what, you know, for analysis. What we rely on is the scientific conventions of citation. So so you would cite this public data set on Dryad when you publish. I mean, that's what the expectation is. Now the next question is, you know, either data like if you need help, if you want to collaborate, there's a number of scientists that are working on this project in a lot of different domains, and we're happy to help. We're happy to develop new projects from it and in a collaborative way. But you know, to the extent that you can make use of this independently, then you're welcome to. Yeah, it's open.
That's a fantastic resource. Like I can't emphasize it enough. I've been working on quite similar phenomic data, but on a different platform, and I didn't realize like how much I could learn just by downloading your data sets and playing around, especially when it comes to like standard practices for how you store your data. Because in our program, I'm kind of making that up as I go. So it's nice to be able to look at how you're doing it and what's working for such a large scale phenomic operation.
Yeah, and I mean, yeah, I'm happy to discuss lessons learned at some point, but I mean, we've gone through different iterations of how to store it, how to organize it, and such. So, yeah. I can't hear you.
You're muted, I think. Maybe. No?
not muted, but we can't hear you, Felipe. the joys of online things. Yeah, so I guess why don't we just start with a general thing of what would you say are your biggest
Can you
lessons
hear me?
learned? Yes.
Yeah.
Hello. Hello. Oh, now it's okay.
Yeah.
Yeah.
Awesome.
Yes.
I'm back. I guess my internet maybe have a break. For example, if I have a data set in my program or someone that is over there watching us now, and they want to put their data on this platform, how they can do that?
Well, that's a great question. So and people asked that quite a bit as we were developing this. So is this going to be a platform for phenomics data? And so we don't currently. It was never designed to be a general purpose platform, but we use general purpose tools, including things like Globus and Dryad and such. And so what I would my first suggestion is generally to say either find a good domain-specific repository or talk to your librarian about how to store it. And the next question is, how can it be stored in a way that can be easily combined with our data sets? So, a lot of the sensor data, we use OGC standard formats like the GeoTIFF in particular and the LAS files, so that it's interoperable with different softwares. And so if you and so that people, so that it's easy for software and for people to figure out where the information they need is. Like GeoTIFF have all of their geospatial information in a particular place, etc. And so so that's my primary suggestion is to work with your local librarian or your domain specific repository. Now that it also kind of brings up this question of the archive data versus the living data. So being able to, so we have all of this data in a database that can be queried through its API. And if you wanted the data that was not curated or public domain, that's how you would access it. For example, for the trait data. But that's not a snapshot in time. So once you're ready to publish, once your data is curated, putting it in a trusted repository, I think is the best place. I mean, as opposed to genomics data, where this is really my opinion though, but I guess informed a little bit from the experience. But genomics data, NCBI is a massive place where you can find all of the genomics data, and that's really fantastic. It's a fantastic resource, but I don't know that that's the best way to go with phenomics data. I think it's institutions can host their own data. It gets very, very expensive to host data. I mean, we've got about a petabyte. I can't remember what the numbers were, but then they're in the hundreds of thousands of dollars per year to host a petabyte of data, and that's as cheap as it can go for that much data. So but fortunately, we've been, NSF has supported a lot of storage at places like Cybers and the National Center for Supercomputing Applications. Yeah. So if that helps.
There were costs associated with putting the data on Dryad?
Well, no, there were not, because the University of Illinois has an account with them, or what, like an institution,
like a sponsor or something.
Yeah. So they can put data up there. I think that for under a gigabyte or two, which is what we've got on Dryad, it's on the order of $100, $150 or so. So, yeah.
Yeah, awesome. Yes, great. Kristina, for example, when we, as soon as I have my data set, you are a data scientist, what the first thing that you do? You do the whole graphic visualizations, or normally, depending on the data set, the first thing that I do is complain. If it's too big, or if there is, I have sometimes when we work with breeding program, we have problems with cultivar names, and you guys probably, because you have many data and time, that can be a really big problem too. So there is any way that you recommend to do a classification for treatments that can be used like more a pattern for breeding populations or experiments?
Wait, so what's the problem with the cultivars? Is it just there's a lot of them, or?
Yeah, each person sometimes put a different name, and this name between programs go changing, but it's the same genotype, you know? So then when we try to combine all this data that you said in the time, then you don't find a genotype that's supposed to be there. And then when you see it's like a comma, it's a slash. Or so how you recommend to identify treatments?
Yeah, so I haven't dealt with this with genomics data, but I've dealt with it with taxonomic identifications of organisms. Yeah, where people will, so some species names change over time, or the like classification of species change over time, and then just like having errors in the data too. And so, yeah, I mean, I don't know. There's an R package actually for dealing with taxonomic names that can help with some of that. It does some sort of automated cleaning. Yeah, I don't know if there's like somewhere that you can pull down like how names have changed over time. Like if there's sort of like track records of like how cultivars have changed. It used to be called this, and now it's called this, depending on like the date it was like put in the database or whatever. Yeah, no, that's really tricky, especially when you're dealing with a lot of data. Like you can't sit there and go through it line by line. You're just inevitably going to lose a little bit of data because you can't get it all harmonized correctly. So yeah, and some of that is a little bit on the data providers, but if you're combining very different data sources collected by different people, like, yeah, it can be really hard. I mean, you kind of sometimes just have to talk to the people who collected the data and be like, hey, you have this like cultivar name in here, but like it doesn't exist anywhere else or something like that. Like, what is this actually? So it can be really painful.
Yeah, I got to do a lot of that, in fact. And, you know, including just having like PI dash and then the number versus PI and then the number, for example. One of the things that we did was we link all of our data sets to a standard repository. And I can't remember where it is, but in the metadata, there's a file called germplasm season4.json. And that has, you know, what we call it, and then what it is on the on the repository, you know, like the canonical source, and then a link to the canonical source of where it's identified. So that's yeah, how we handled that.
Yeah, it seems like you'd have to have that done pretty individually, because if you were working for you know, a breeder who's actively producing lines, then you get all of those names that have lots of zeros and dashes and all of those things that can be very problematic in your end data.
Yeah.
All right, Claire, do you have more questions?
It's a good question.
I do have one last question, but I'm keeping this for the end.
So Claire, I think you were asking about lessons learned at one point, and I can say that I actually kept a Google Doc of lessons learned, and there are a lot of lessons that we've learned in a lot of different areas. I mean, starting with the fact that when the machine was provided, it was, you know, the idea was that it was going to be installed and it would be generating phenotypes. So the company Lemnitech that installed it, the way that the request for proposals was written was that they were going to, you know, have the entire pipeline written, all the calibration done and everything, so that we would have something to compare what we're doing to. What ended up happening, what we ended up getting was a gantry with the sensors installed, and we could get the raw data files for the sensors, but there were none of the even the sensor companies that they have their own software for processing their data. None of that was provided, so we had to start from scratch, and in the process learn a lot about calibration of different instruments and how to use them and under what conditions they work and what they don't work. It's the hyperspectral camera in particular is very difficult to calibrate when you've got a big white box in the middle of the summer sun, half shading the plot. And so so there were those lessons learned, like making sure that expectations are clear, and then just dealing with all of this data. Another thing that we learned was like the reason that we've got out of 9 seasons for the over the course of the TERRA REF project, we've got 2 that are published. It's because we realized in order to get something out there that was useful, we were going to have to really focus on something that was of interest and get that out there. So that, and the idea is that as people use this and they get interested in other, other populations of sorghum, or we also have there is wheat trials every winter. Then we can work with people to make that data available. For example, we're working with the wheat head imaging data set group to provide some of the images that we have of wheat heads, for that contest. So
Nice. Yeah, great. Yeah, I don't know if you know, but I'm gonna start working with wheat now.
Oh, cool. Okay.
Yeah.
If you need any data, we've got it. It's
Awesome.
it's Durham wheat. So one person was disappointed because Durham wheat is different, has a different ploidy, but yeah, it's what it is.
Yeah, great guys. And yeah, my last question is mainly, let's see, Kristina, Python or R, and why? I'm a R lover.
I don't want to start like any flame wars or anything here. That's not the intention. Yeah,
it's a joke.
like
it's a joke.
Yeah, the real answer is use the tool that works for you. Use something that other people around you are using. So yeah, it's funny though. When I first started learning how to program in grad school, I was learning R and Python simultaneously, and I used them both for one project. But I kind of used them backwards. I did the data cleaning and stuff in R, and then I did the visualization in Python. And now in hindsight, looking back, I totally did it backwards. So it would have been much faster and easier if I'd sort of used the right tool for the right job, but I didn't know at the time. So yeah.
Yeah, thank you. Yeah, so.
But I love R too. Go on.
Yeah, R lovers. Yeah, so guys, yeah, any other final comment for our workshop today?
Oh, we again do have the notes for everything that we went through. I don't know if they're in the YouTube description, but maybe they could be added just for folks who want to refer back to what we actually did.
Awesome.
Yeah, I don't think we
or
have
just
them in
watch
there
the whole
now.
video.
Add them.
Okay, that would be helpful.
Yeah,
Maybe.
so thank you so much for your time, for being here with us, for sharing everything with us, your knowledge, the great work that you guys doing. So hope see you guys here soon with more new stuff to share. And thank you.
Yeah, thank you so much. This is just such a fantastic thing that you're doing with PhenomeForce. Thanks for having us, and thanks for doing this for the community. So.
Awesome.
Yeah, Neil did a great job setting this up.
Awesome. Thank you, guys. Bye-bye.
Okay, bye.
Yeah, Claire, let's present our next workshop, which is gonna be in 1 week for now. Let me open here. So next Friday, we're gonna have, you're back, Claire? So next Friday on 23rd, at 10 a.m. same time here at our channel, we're gonna have this other workshop now with our colleagues from France, and they're gonna talk about RGB depth plant phenotyping from top view, system and image analysis. And we are really excited to have this workshop as well. And as you can see here, we just had our second workshop of this season, and we are planning to have our next seven workshops, yes, from now. And please, if you are interested to be part of this project, contact us, go in our social media, and it's really great to have you guys here. Are you there, Claire?
Can you hear me?
Yes, now I can hear you.
Okay, sorry, my headset died, but yes. No, I'm looking forward to the next few sessions, and hopefully if you're interested or you think it might be of interest to a colleague, make sure you share the link so we can spread this great information from our wonderful speakers who volunteered to come out to do this.
Yes, awesome. And again, everything is open, everything open source, so we need your help by signing here and also sharing the videos in your social media and helping us to put the information out there. Okay? So see you next Friday. Bye-bye.
Yeah, next week we'll be back.
Bye.